Hello everyone. Welcome to LifeScience Simplified. Here, we make learning simple, easy and quick, helping you grasp complex concepts in minutes, perfect for learning fast and revising smarter. Before we dive in, make sure to subscribe and hit the bell icon so you never miss our upcoming videos. If you are preparing for CSIR NET Life Science December 2025, today's session is going to be a complete game changer because we are diving deep into Unit 1 molecules and their interaction relevant to biology, commonly known as biochemistry. This is one of the most conceptual, numerical, and high weightage units that can literally decide whether you qualify JRF or not. So stay till the end because I will be covering topic-wise analysis, most important or not to skip subtopics. How to study this unit effectively, how to prepare from MCQ to theory, and a complete smart strategy to crack this unit. So, grab your notebook and let's simplify biochemistry together. Part 1. Importance of this unit. Let's start with the big question: why is this unit so important for CSIR NET Life Science? Well, Unit 1 molecules and their interaction relevant to biology contributes around 17% in Part B and 8% in Part C, but here is the catch: these questions are not factual, they are conceptual and application based. So if your fundamentals of biochemistry are strong, you can easily attempt not only this unit but also questions from enzymology, metabolism, and even cell biology. And the best part, this unit connects directly with Units 2, 3 and 5 of the syllabus. So, once you master this unit, it will create a strong base for almost 25 to 30% of your entire paper. Part 2. TOPICWISE analysis. Now let's break down this entire unit topic by topic exactly as given in the syllabus. We will discuss what each topic means, how many questions you can expect, and what to study within it. Molecules and biomolecules. This part forms your foundation. It covers atomic structure, chemical bonds, biomolecules, carbohydrates, lipids, proteins, nucleic acids, and vitamins. From the exam point of view, focus on functional groups, acid-base chemistry, isoelectric point of amino acids, and properties of peptide bonds. Sometimes you get direct conceptual questions like, at which pH will a given amino acid carry no net charge? So revise pKa values, peptide bond characteristics, and Switterian concept. Stabilizing interactions. Now this topic is super important because CSIR loves to ask about non-covalent interactions. Here you need to study hydrogen bonding, ionic and van der Waals forces, hydrophobic interactions. These forces decide how proteins fold, how DNA remains stable, and how enzymes bind substrates. Tip: practice identifying which type of bond dominates in different biomolecular structures, for example, hydrogen bonding in DNA base pairing, hydrophobic effect in protein folding. This conceptual clarity will help you in both Part B and Part C questions. Principles of biophysical chemistry. This is the numerical section of the unit. Focus on pH and buffer, Henderson-Hasselbalch equation, reaction kinetics, thermodynamics and delta G colligative properties like osmosis, diffusion. CSIR loves asking numericals like, calculate delta G for a given reaction using delta G is equals to minus RT natural log of K, or find the pH of a buffer when base to acid ratio is given. So, make sure you understand formulas, not just memorize them. Practice small numericals daily, it builds speed and confidence. Bioenergetics and energy transduction. This section is a CSIR favorite. It covers glycolysis, TCA cycle, oxidative phosphorylation, chemiosmotic theory, ATP synthesis, coupled reactions and redox potential. Most Part C questions are data interpretation based, like, given redox potentials, predict the direction of electron flow and ATP yield. So, understand how energy is transferred from one molecule to another and memorize ATP yield charts. Enzyme catalysis and kinetics. This is the most important section, the king of biochemistry. You cannot afford to skip this one. Study in detail Michaelis-Menten kinetics. Lineweaver-Burke and Eadie-Hofstee plots. Km, Vmax, Kcat. Types of inhibition: competitive, non-competitive, uncompetitive, allosteric regulation and feedback inhibition. Every paper has 2 to 3 direct questions from enzyme kinetics, either graphs or conceptual numericals. Tip: practice graph interpretation. For example, if Vmax stays the same but Km increases its competitive inhibition. Learn these patterns visually, not just theoretically. Conformation of proteins. Next, protein conformation. Here, you will study Ramchandran plot, secondary and tertiary structures, domains and motifs like helix turn helix, zinc finger, leucine zipper, protein folding and denaturation. CSIR sometimes gives a plot and asks, which amino acid combinations are allowed in this region? So revise phi and psi angles and understand the logic behind allowed regions. Conformation of nucleic acids. This topic covers A, B, Z DNA, RNA structure, tRNA, microRNA base pairing, major and minor grooves. You might get a conceptual question like, which form of DNA is left-handed and rich in GC pairs? Answer: zDNA. Simple, but conceptual. Stability of proteins and nucleic acids, closely linked with previous topics. Focus on denaturation, renaturation, melting temperature, Tm, role of urea, SDS, pH, and heat. Factors affecting double helix stability. Metabolism. The last part: metabolism of carbohydrates, lipids, amino acids, nucleotides, and vitamins. Study glycolysis, TCA, beta oxidation, urea cycle, nucleotide biosynthesis. Regulation of pathways. Role of cofactors and coenzymes, NAD, FAD, coenzyme A, biotin, thymine, etc. Focus on rate-limiting enzymes and energy yield. And make summary tables like glucose yields 38 ATP or palmitate yields 106 ATP. Most important and not to skip topics, let's quickly summarize the must-do, high-yield areas. Enzyme kinetics and regulation, bioenergetics and oxidative phosphorylation, protein structure and folding, thermodynamics, delta G, delta H, delta S relations, pH, buffers, and Henderson-Hasselbalch metabolism of carbohydrates and lipids. Stabilizing interactions, hydrogen, hydrophobic, ionic, DNA conformations and tRNA structure. If you cover these 8 areas solidly, that's 90% of your CSIR NET biochemistry weightage sorted. How to study this unit effectively? Alright, now let's talk strategy, how to actually study and retain this unit effectively. Step 1. Build strong concepts. Study from Leininger's Biochemistry, its gold standard. For basics, refer Satyanarayana. Focus on why Km changes, why delta G is negative, why hydrophobic effect drives folding. Step 2. Make concise handwritten notes. 1-page notes per topic. Include only key formulas, definitions, and examples. Keep them ready for last 10-day revision. Step 3. Practice MCQ. Solve 10 years CSIR PYQ attempt 20 MCQ daily. Analyze mistakes. Focus on graph-based and numerical questions. Step 4. Data interpretation. Practice enzyme kinetics and buffer numericals. Understand metabolic regulation questions. Revise using flowcharts and visuals. Step 5. Quick revision. Revise daily, 1 topic in morning, 1 in evening. Prepare a formula and pathway chart. Attempt full Unit 1 mock test weekly. So friends, that's a complete breakdown of Unit 1 molecules and their interaction relevant to biology for your CSIR NET LifeScience December 2025 exam. If you study this unit strategically, concept clarity from books, daily practice of numericals, PYQ analysis, and weekly revisions, you will not only master biochemistry but also strengthen 3 to 4 other units automatically. Remember, CSIR NET does not test mugged up facts, it tests your logic and understanding. So study smart, not hard. If you found this analysis helpful, don't forget to like the video, Subscribe to LifeScience Simplified and turn on notifications for more upcoming CSIR NET strategy videos. I will see you in the next session. Till then, study smart, stay consistent, and keep learning.